Dear researchers, student colleagues, and community members, welcome to the Graduate School One Health Emerging Infectious Diseases webinar series. I am Paola Gamboa, Education and Digital Manager of the Graduate School. Thank you for joining us today. We are very glad to have you with us in the One Health and Emerging Infectious Diseases Social Sciences webinar. I am pleased to introduce Jules Villa, Chair of the Social Sciences Webinar Series and next academic year teaching unit. Jules Villa works on the social and historical dimensions of health emergencies and the responses they elicit. He has largely studied the Ebola and Mpox epidemics in the Democratic Republic of the Congo, a country he has visited regularly since 2019, and he has taken part in several response activities against these epidemics. He joined the Anthropology and Ecology of Disease Emergence Unit in June 2024 as a postdoctoral researcher with funding from the Agence nationale de recherche sur les sida et les maladies infectieuses émergentes. His postdoctoral research project at the Institut Pasteur is a book focused on the history of Mpox in the Democratic Republic of the Congo and on the ways in which these epidemics have been studied and fought over the time. In today's session, the last of our academic year, we are very happy to welcome Emmanuelle ROTH. Before we get started, a few quick reminders. Today's presentation will be in English, but don't let that stop you from asking questions in either English or French. Feel free to post your questions in the chat or raise them directly during the discussion session. When asking your questions, we would like to know a little more about who is behind it. Please introduce yourself with your name, your role, researcher, student, or otherwise, your institution and the country you are joining us from. It is always nice to know how far our webinar travels. Thank you all for being here today and enjoy the webinar. I won't keep you waiting longer. Jules, the spotlight is all yours.
Thank you so much, Paola. Hello, everyone. The first thing I want to say is that I'm very thrilled that we have Emmanuelle ROTH today with us, but also that I wish we were doing this webinar in another circumstance, because when we planned it, obviously we couldn't foresee the current Ebola situation in DRC that we are all Ebola researchers, following closely. So a quick introduction for those of you who do not know Emmanuelle ROTH. So Emmanuelle ROTH is an anthropologist currently affiliated at the Rachel Carson Center in Munich as part of the ERC-funded project Fragments of the Forest. Emmanuelle's work focuses on health and sciences. After initially training in humanitarian action, she became involved as a social scientist in outbreaks of Ebola in West Africa in 2014-2016. She then moved to academic research and wrote a PhD thesis at the University of Cambridge titled The Truth About Ebola, Insecure Epistemologies in Post-Break Forest Guinea, and defended in 2022. Her postdoctoral research additionally draws on environmental history, and resituates the virus hunting endeavors she described in her thesis at the crossroads of natural history, conservation science, and infectious disease research. Emmanuelle currently investigates shifts in understanding, caring for, and valuing bats through examining practices of collecting bat specimens and samples in Mount Nimba and Upper Guinean forest, but she'll talk us through this. So Emmanuelle, the floor is yours, and thank you very much again for accepting our invitation.
Great. Hi, everyone. I want to start by thanking Jules and the Graduate School of One Health and Emerging Infectious Diseases. It's a real pleasure. I'm very delighted to be presenting my work to you today. And so what I will be doing is presenting some material from a book manuscript that I'm currently finishing, which is based on my PhD thesis in anthropology that I completed about 4 years ago. The book is titled Echoes of Ebola. Oops, I cannot. How do I? Oh, there you go. The book is titled Echoes of Ebola, Virus Hunters and the Making of Insecurity in Guinea. and it's a book that's under contract with the University of Chicago Press. And I apologize, I'm very much going to be reading my script. I'm going to try to make it as lively as possible, but that's written talk today. So what is the book about? The book explores the realities of virus hunting in the global south. It tells the story of virus hunters, the animals they collected, and the truth that echoed in West Africa around the search for the origins of Ebola after the deadliest epidemic of the disease in West Africa. So it's very much a story of One Health, which is why I'm happy to present to you today. And I also didn't foresee that it would be speaking to a very sad actuality. Since the 1990s, you may be familiar with depictions such as those of brave scientists braving the dangers of Africa for the welfare of mankind. This is very much so. Here you see Nathan Wolfe. You may know Peter Daszak. Here is Bill Gates. Those are kind of heroes of our pandemic imaginary, obsessed with tracking the germs that swarm in the jungle and bringing them to a lab. The pandemic of COVID-19 has updated the figure of the virus hunter and turned them into a hero of global health who uploads viral sequences to open access databases and prevents outbreaks with equations. That's the promise, at least. The message is very clear. Virus hunters help us anticipate viral threats and ultimately make the world safer. But in the caves where the dirty work of bat sampling happens, unknown threats are overshadowed by real dangers. Virus hunters must find their bearings in unfamiliar terrain, navigate instructions from contractors, and sidestep the clamors of people who suspect them of spreading viruses. So my book draws on media analysis, the scientific literature, and 18 months of ethnographic fieldwork in Guinea between 2014 and 2023, and explore the scientific practices, discourses, and performances of a group of Guinean veterinary doctors employed in the aftermath of the largest epidemic of Ebola that caused major disruption to West Africa between 2013 and 2016 and caused over 10,000 deaths. Let's go back to what's epidemic surveillance. So epidemic surveillance is underwritten by the idea that you have to integrate distant places into a global system of monitoring, recording, and intervention. So to that end, you need data that is shared in real time on open access platforms. You need close relations with NGOs, with country authorities to intervene very quickly. Detection has to happen early, results needs to be shared transparently, public health response needs to be fast. But that surveillance is operated by people that this book is concerned with, and those people are concerned with much more proximate threats than the next pandemic. Epidemic surveillance may rest on this fantasy of immediacy, but it's very much a hyper-mediated product whose first line rests in the end on human workers. And managing and preparing for epidemics is sustained by these people who remember past epidemics and experience everyday hazards. So the labor of securitizing the world against pandemic interferes with these hazards. So the book makes the argument that insecurities arise not just from the uncertainties that surround novel viruses. Insecurities also permeate the labor of workers employed to prevent the next pandemic. And so global health security depends on anticipating insecurities and anticipating them correctly, including those it generates. This is an outline of the presentation I'll be giving to you today. I'll return quickly to the book at the end. And here's a map to just situate this talk. So you see Guinea in red on the map. The region circled in red is Forest Guinea, where the Ebola outbreak of West Africa supposedly started. The arrow points you to Meliandou, the supposed place of the initial spillover, which is very close to Gekedou, which is the place where I now want to take you to a night of November 2018. That's the evening, but the scene happens in the night, past 1 o'clock in the compound of the Direction Préfectorale de la Santé. This location that you see was a favorite of bat virus hunters in Guinea because it had old colonial buildings that hosted a lot of insect bats. So they were very easy to capture and sample on the spot within the safety of the Direction Préfectorale de la Santé, away from prying eyes. A rickety plastic table stood on a cement slab that you see here, which was cast by Médecins Sans Frontières Belgique. The slab was a relic of the tents where the Ebola patients were quarantined and cared for in 2014. Only empty water towers and a broken medical incinerator remained of this time of emergency. In this place, which you see here, 5 workers directed their attention to bats, captured only minutes before, which they moved along the assembly line of the lab work. Those efforts, the PREDICT program hoped, I will return to the PREDICT program, would help identify the origin of Ebola and reduce the risk of a future outbreak. But those workers did not call themselves virus hunters. They were just, to them, préleveurs, or samplers. One by one, each animal was pulled out of a tissue bag by the scruff of the neck after being weighed. Its sex was determined, age evaluated, forearm measured, and species guessed. Then one sampler carefully turned the animal over in his hands so that another one could extract 9 samples from its mouth, its anus, and its brachial vein. A secretary ticked boxes on a spreadsheet for every sample collected. Meanwhile, a fourth sampler meticulously collected them in tubes, which they arranged in a numbered cryobox. The beams of a spotlight projected their moving shadows on the walls of the Direction de la Santé. Hey, can't you hear me? I need a paper towel now. Michel yelled. Michel, who sat at the end of the line, introducing capillary tubes into the blood separation centrifuge to isolate the bat serum. He was shouting for help to wipe the sweat that had condensed on his face shield and was clouding his vision. The hazmat suit, hazmat as you see here, was sticking to his sweating body. The humming of insects attracted by the spotlight and the roaring of the engine muffled his voice and the squeaking of bats. Michel struggled with the centrifuge, which always stopped spinning short of the 2 minutes necessary to fully fraction the blood and had to be restarted. The other 4 samplers, drained and hurrying to disinfect the equipment and pack up the lab, were arguing at the other table. Who the hell had forgotten to load the sharps containers where used needles need to be disposed of? Could they safely throw them with the remaining refuse? Meanwhile, the voice of their supervisor resonated in Michel's head. 11 bats should mean 22 serum tubes. What will I tell the Americans? Michel had been scolded earlier for breaking some of the tubes while restarting the centrifuge. The defective machine prolonged their work hours into the tropical night. Why not sample the animals in the morning light? It would have certainly made that labor very much easier. But the animals, the bats, were only to be detained for up to a few hours, because if you put them in a bag, it can injure their wings. It causes stress, which depletes their energy. So extended detention really compromises their immunity, their flight, and then exposes them to predators when they are released. The samplers were told to treat the bats humanly, and so they tried. Such a field laboratory where sentinels of emerging infections are to be treated humanly is a recent contribution of the One Health approach. And it's also a requirement that the project sought to impose, which had consequences for both humans and bats, which we'll go into. So let's go back to this idea that epidemics of zoonotic diseases such as Ebola have a spatial temporal origin in a so-called reservoir, an idea which very much anchors these queries into their origins in places where they start. And this place was Guinea in this case. Scientists have been investigating outbreaks of Ebola since 1976 by sampling animals in outbreak sites with the hope of detecting traces of the virus in some of them. In 2005, French virologist Éric Leroy found viral RNA in fruit bats captured at the border between the Republic of the Congo and Gabon. And from then on, so before then, scientists had been kind of like haphazardly sampling guinea pigs, primates, insects. From then on, they really focused on conducting codified bat roundups. Yet this hypothesis of a bat reservoir is very much contested until now, both on ecological and epidemiological grounds. There is no certainty on the species that maintain the disease in between epidemics, which justified this work in 2014. After 2014, rather. It's in 2014, when Ebola failed containment measures in West Africa, that the American aid agency USAID extended the PREDICT project to the region. PREDICT was the largest and most well-funded endeavor to proactively secure the world against pandemic threats. It benefited from more than $200 million over 10 years. It had a very straightforward mission, establishing the most comprehensive database of viral genomes. And the most threatening viruses they expected to find in the wildlife of areas deemed at risk of epidemic spillover, the so-called hotspots located as if by necessity in Asia, South America, Africa, and identified, I cannot go back, yeah, so hotspots that were often represented using this sort of aerial blood stained maps. And PREDICT was not so much focused on mitigating the spread of viruses after they emerge in human populations, which was labeled a reactive strategy. They rather wanted to reduce the opportunities of viruses to transgress divides between animals and humans, which is this like program of viral forecasting, which PREDICT was very much kind of a forerunner and heralded as pushing forward. In the 21st century, as pandemics drove home the urgent need to know more about how novel diseases emerge, scientific fieldwork became the stuff of protocols that police work that challenges boundaries between people, animals, and pathogens. The One Health agenda was being mainstreamed across international organizations, and research strategies increasingly combined biosecurity, focused on the risk of disease transmission, and bioethics, attentive to questions of animal welfare. In Guinea, PREDICT thus elected to work with veterinarians. And you see the organization that were involved with PREDICTs, a lot of conservation organization and biosecurity, biosecurity organizations. Vulnerabilities in the plural is a keyword of the One Health approach. The goal is to find a balance that takes account of vulnerabilities of both humans and animals. So pandemic anticipation is essentially a balancing act. But like any balance, it always threatens to tip. Professionals from the global south seem to excel at these gymnastics of preparedness. Like Michel, they seem to have a talent for performing biosecurity and animal welfare in demanding circumstances, which One Health institutions are glad to tap into. This is highlighted by recent news media coverage on African virus hunters, and we'll now take a quick detour through the visual imagery of pandemic prediction before returning to that evening scene in Gekedu, where we opened this presentation. Some of you may know Gaël Daran Maganga, a veterinarian and virologist from Gabon, one of the most visible public faces of bad virus research in Africa. And since the West African Ebola outbreak, Maganga has starred in a lot of interviews, documentaries, reports. He very much exemplifies the figure of the African virus hunter. Black scientists, technicians, and wildlife workers appear as both skilled professionals and resilient laborers in these reports. They are portrayed as capable of carrying heavy equipment through difficult terrain, negotiating local customs, caring for wildlife, and confronting infectious danger. Because they can improvise in difficult circumstances, Black field workers are guardians of health. And there is truth to this image. Science is indeed, as Crystal Byrock writes, more of an improvised art than a scripted thing. There would be no global health science without the embodied skills and practical knowledge of field workers. And there's been a lot of scholarship on how this research relies on workers whose labor affects knowledge of what makes science possible. And the machinery of global health is beginning to acknowledge this. So here's a picture, for example, that was very much used by PREDICT. that celebrated field workers as the champions of One Health. The difficulties of fieldwork are increasingly recognized and also dramatized, as you can see on this picture, as evidence of flexibility and sacrifice. Yet, back in Gekedu, Michel's call for help went unheard as he tried to calculate how much faster his colleagues should fill tubes with blood so that he would have enough time to centrifuge them before the generator ran out of fuel. What if, instead of focusing on the visual spectacle of virus hunting, we attend to the sounds of this labor? Listening reveals that samplers were not simply improvising around bats, they were continually sounding out many different vulnerabilities, those of bats, certainly, but also those of equipment, infrastructure, livelihoods, bodies, and professional identities. Their work depended on assessing what might fail, how severely, and for whom. So the narratives about African improvisation risk romanticizing adaptation to scarcity. This talk takes such improvisation seriously, not as an innate cultural capacity, but as labor. Predict samplers did not simply react to uncertainty, they expected uncertainty. So rather than depicting preparedness as a choreography balancing risk and care, I show how virus hunting depends on workers who continually navigate multiple insecurities, often without a resolution. A balancing act. Capture. Just an hour in Gekedu before Michel tried to stabilize the wobbly lab table, a call resounded. Capture. This was the sampler's way of signaling that an animal had been caught and that the staff meant for lab work that night ought to get ready and don protective personal equipment, or PPE, to collect the creature before it got too tangled up. But they should not arrive too early either, lest they stay in PPE for too long and begin suffocating. It all depended. Could they expect to catch many bats from the roofs of the Direction de la Santé on that cool, dry night? They had to assess that likelihood and many other unknowns. Although the donning and doffing of PPE had become routine by them, samplers were admonished to maintain their concentration. As a Predict veterinary epidemiologist repeated during a training session, the most important part of PPE is not the hazmat suit, it's not the respirator, it's your brain. Use your brain, Nathalie urged Omar, Michel, and Norbert as she read aloud, as always, a sheet detailing each step. The three men stepped into oversized hazmat suits and adjusted the elastic over their rubber boots. They faced some disposable face shields to protect themselves from bat urine and blood. Norbert and Michel placed N95 respirators over their noses. Then they tied plastic aprons around their waists. Finally, they put on nitride gloves. Norbert even added a second pair. Now they were ready. The Predict protocol estimates that the prevalence of pathogens, of harmful pathogens in bats is between 0.01 and 1%, which is quite small. But samplers were taught to observe very strict biosafety precautions to protect themselves from bats and their bodily fluids. Bats have sharp teeth that can easily pierce several layers of glove, which is why some workers, like Norbert, preferred as many layers as comfortable. But a balance between the safety of both the field personnel and the bats being sampled, and here I quote the protocol, was sought because bat research also generates risks for bats. The sampling protocol prescribed using the most humane and least invasive techniques to sample wildlife while minimizing pain and distress. I quote the protocol, in accordance with this idea of a One Health surveillance. Bats were to be captured, sampled, and released on the spot. Sampling had to be ethical and safe. No more than 1% of the body mass in blood could be taken. They couldn't use anesthesia because it can be bad for the animals. Animals should not be stressed. They were to be released within 1 kilometer of the site of capture. So all these principles, the samplers of care for the animal, the samplers collapsed them into a mandate. We were told to treat animals humanly. And so they were given a workshop To teach them how, they learned there to use a guide for identifying species, a bat detector. They learned to attune their senses to another species. We were told to treat bats humanly became kind of a mantra, always expressed in the passive voice. We were told to treat them humanly. And through that passive voice, staff acknowledged the limits of their own agency against the authority of the institution that issued the command. Let's get closer to the laboratory table now. For each bat, sampling required 10 to 15 minutes of handling tubes, disinfectant, scissors, swabs, and animals that struggled to escape. Although everyone had been trained to handle bats, restraint was generally entrusted to those most confident in accomplishing the task without being bitten or injuring the animal. So samplers negotiated with the protocol. Omar, for example, refused the leather gloves, a pair of leather gloves, which you can see here, which normally you have to wear when handling fruit bats, whose bites are deemed more dangerous. But those gloves, they also reduced sensitivity, and so you risked injuring the animal. So Omar, who was a forest warden and claimed to have an affection for wildlife, preferred to rely on his dexterity alone. Anyways, many bats died in the early stages of the project. Even the supervisor accidentally killed one while attempting to sample it. He was distressed by this breach of protocol, and he immediately phoned the country director to report the incident. But after hanging up, he turned to the team and he added mischievously, "Mes condoléances." Months later, that story still provoked laughter among the samplers. Humor helped release some of the anxiety generated by this balancing act they performed every night, such as the anxiety of losing one's job. Over time, samplers became skilled at anticipating uncertainty, weighing different risks, and fine-tuning, negotiating with scientific principles. But this joke, mes condoléances, when killing a bat, is an invitation to understand that these calculations did not stop at biosecurity and animal welfare. They also considered waste disposal, infrastructure, practicality, comfort, and professional experience. This labor of forcing multiple risks mediated the contact and the distance with bats. Some people, like Omar, judged their skills sufficient and dispensed with leather gloves, but other contexts, talents, and affects could produce different human-bat encounters. One night, as the team was preparing to leave a capture site, a woman called out, "Please, help!" A bat had fallen inside her house. While the male samplers dismantled nets and packed equipment, Natalie heard the woman, Natalie, whom you see on that picture, and followed the woman indoors. Natalie described herself as someone who loved bats, and there were not a lot of samplers who loved bats. She marveled at their teeth, which were always white and clean, despite never being brushed, she said. She delighted in the realization that many of the fruits she enjoyed existed thanks to bats and their role in pollination. Inside the house, she found a tiny insect bat, probably a juvenile, clinging to a wall. Red fire ants crawled across its body. The animal had likely struck something during flight and been attacked on the ground. Carefully, Natalie picked the ants off one by one before slipping the bat into a bag. The woman thanked her. Natalie smiled back without a word. She had entered the house without full PPE because she knew that PPE could alarm residents and provoke fear. The juvenile seemed too weak to bite, and she also doubted that samples could be obtained from it anyway. But if she released the bat immediately, it would become prey to a snake, or people would think that she was trying to release diseases in their houses. So she decided to bring the animal to the lab, give fruit juice as recommended, and release it later. With careful gestures and a smile, Natalie hedged dangers and future reproaches and recreated the role of the female virus hunter. She was the exception. The work of restraining bats at the sampling table remained almost exclusively male territory. That division of labor points to another vulnerability that interfered with choreographies of care and security, threatened masculinity. Bats were gendered by field workers indeed. They became the target of jokes that further feminized them, such as chauve-souris, which rhymes with the French nourrice, used in West Africa for nursing mothers. Samplers also frequently commented on bats' ugliness, which kind of reinforced associations with malice and cunning, which are already present in the regional folklore. Affectionate gestures towards bats, like warming bats, petting bats, expressing sadness at their death, were often mocked and exaggerated. Fieldwork heightened the need to maintain distance with these animals. Samplers were mostly educated men from towns. They wore city shoes, perfume, and shirts to community meetings. They played music loudly from smartphones to signal their prosperity. During the Ebola outbreak, many had struggled economically, and so now they had employment in a project which promised respectability and financial stability. But fieldwork constantly undermined that image. Their clothes became dirty, the sleep was scarce, evenings ended in exhaustion. And instead of caring for valued livestock, those vet doctors were spending their nights capturing bats, which a lot of locals considered repulsive. So their sense of masculinity was unsettled, and the unease surfaced in banter. Technical expertise and the division of labor thus did more than manage disease risk and animal welfare. They also helped establish distance between field workers and bats. Through adjusting that distance, socialized, gendered, racialized species, samplers fashioned identities for themselves. They were not simply experts balancing biosecurity and bioethics. They were precarious workers undertaking forms of care that were feminized and uncomfortable under conditions shaped by numerous constraints. And samplers would learn a lesson about the necessity of probing and maintaining this distance with bats in relation to yet another distance that's separating them from their bosses, which all went into this work of improvisation that I'll describe now. Another scene from Gekedu. That night, representatives from USAID undertook a field visit to observe PREDICT at work. The team was sampling in a village that night amid residents whose curiosity had been aroused by the presence of foreigners and the unusual activity. When the bat was netted, the supervisor himself stepped in to demonstrate the procedure. He slipped on a pair of gloves, but he was hasty to untangle the animal. Maybe he also feared suspicion from locals, so he did not wear the remaining PPE. Meanwhile, the USAID visitors enthusiastically photographed him working with bats, and a lot of pictures were taken during these nights of work. Some of these pictures were then sent to PREDICT's global leadership, and the reaction was swift. The entire Guinea team incurred the anger of managers who worried about the biosecurity failures visible in the images. He was wearing a lot less PPE than on that picture that I show here. For several days, staff feared that the incident might jeopardize the project itself and with it, the source of income. Prudence was indicated when it came to disclosing the conditions in which sampling actually took place and those improvisations that they were forced to take. Workers must continually assess what might fail and with what consequences, but they must also evaluate how their own adaptations will be judged by management. Will an improvisation be celebrated, or will it be condemned as recklessness? When samplers adjusted protocols to accommodate unreliable infrastructures, such as the population's mistrust, they did not merely react to absences, they had to anticipate the risks created by their adaptations themselves. Flexibility was a work of feedback. Let us now delve a little further into the labor conditions of virus hunters. Being recruited by PREDICT was serendipitous for Guinean veterinarians whose livelihoods really depended on improvisation. Let's be clear, the motivation for joining the project was primarily financial. There are not a lot of employment opportunities for university graduates, and projects are very attractive positions. Yet many of these samplers were disappointed to be hired as consultants. Their contracts were very short. Rumors circulated that money disappeared somewhere along the administrative chain. No one could prove it. But what mattered was that the samplers perceived a significant gap between the risks they assumed and the rewards they received. So they talk of self-sacrifice. And this did not only refer to wages. Sampling expeditions commonly lasted 9 days. Accommodations were basic. There was no running water, no electricity. Team members slept 3 to a bed or on the floor. They only ate rice for that time. They went to sleep after 1 o'clock in the morning and rose before 7. There was little time for rest. So when samplers spoke of sacrifice, we need to hear echoes of the Ebola epidemic as well, because many of them had participated in outbreak response efforts, and they felt that such work received very little recognition. And although they like to describe themselves as on the terrain, their usual professional activities as veterinarians rarely required prolonged stays in remote villages. Their education rather inclined them towards urban middle-class lifestyles. The projects that promised financial security and prestige, but the reality was very difficult working conditions and not a lot of recognition. Now, working with bats intensified these tensions. Bats are nocturnal animals and are least disturbed when handled at night. Early in the projects, they started sampling the animals the next morning to work in daylight and rested. But the trainers insisted that bats should be released on the same night they were captured, and holding them too long in bags could cause stress. So as a result, sampling began as soon as the first bats entered the nets, a decision which protected bats, but imposed costs on humans. Samplers often put on PPE around 8 in the evening and wore it until the early hours of the morning, which caused exhaustion, heat stress. They worked in darkness, so they sometimes encountered snakes. They worked under artificial lights, which also attracted mosquitoes. And so towards the end of the shift, field workers were exhausted and a lot of arguments erupted. Everyone wanted to finish, disinfect equipment, eat dinner, and go to sleep. As you remember from the vignettes I opened this talk with. Samplers quickly realized that if they wanted to disclose something of these labor conditions to management and to work towards improvement, they should not necessarily frame these arguments in terms of worker welfare because of that rhetoric of selflessness and adventure. Appeals grounded in the ecology of bats proved a lot more effective. How, they asked, were they expected to meet capture targets during the rainy season when bats sometimes remain in their roosts? Would they have to stay awake all night waiting for animals that never appeared? Eventually, capture goals were revised by scientists and staff were reassured that their safety mattered more than the number of samples. But the logistical constraints of working with bats could be somewhat discussed openly. But the affective dimensions of that labor and the consequences were more difficult to articulate and had to be improvised around. I realized this one night while observing another surveillance team. There was an elderly veterinarian named Cissé at the lab table, and when Cissé opened the bag, he discovered a fruit bat that was lactating its pup that was breastfeeding. It's not so clear on that picture, but this is a breastfeeding, lactating bat with a baby. Immediately, Cissé urged his colleagues to work quickly because mother and pup should be kept as little time as possible separated. Pregnant and lactating bats really occupied a special place in the moral imagination of many samplers. They may mock ordinary bats as ugly, but mothers were viewed differently because they were caring creatures whose attachment to their offspring inspired admiration. Cissé attempted to collect a saliva sample from the mother, but the bat suddenly escaped his grip and bit his hand. His scream cut through the night. He shook his arm until the animal released its hold and flew away, leaving its pup behind. A dispute immediately broke out over responsibility for the accident and over the fate of the orphaned pup. Those orphaned baby bats elicited genuine sympathy among field workers. Teams sometimes attempted to attach them to other females, hoping that they would become adopted. But Cisse's concern was elsewhere, and he did not attempt to save the pup. "My health comes first," he said. "I don't have an insurance if I get sick." His anxiety reflected more than immediate injury. During the Ebola epidemic, hundreds of West African healthcare workers died, while the few foreign aid workers whose lives were at stake were evacuated abroad for treatment. The staff of PREDICT had been vaccinated against rabies and toxoplasmosis. The project assured them that they would get booster shots if they were bitten by bats. But of course, there was no vaccine against all strains of Ebola, against Marburg, or many other poorly understood viruses that you may encounter in bats. Officially, their contracts included health insurance, but in practice, most assumed that it would require a lot of paperwork, and so they paid their medical expenses out of their pockets. And so whenever a bite occurred, the first concern was whether the teeth had penetrated the glove. Then the worker would monitor his body anxiously for signs of illness, and most bites went unreported. The staff feared being blamed for breaches of protocol, for improvisations. They worried that repeated incidents might jeopardize future contracts. Silence often appeared safer than disclosure. Infrastructures form the background that allows scientific work to happen. Medical anthropologists have long noted that healthcare workers operating in under-resourced settings routinely compensate for missing equipment and power cuts through improvisation. Making do like this is not exceptional. It's really part of the labor that sustains medicine and science. Scientific field workers adjusted not only to bats and pathogens, but also to fragile infrastructures, precarious employment, managerial expectations, and fears about their own futures. They were not simply masters of improvisation who react to absences. Their improvisations were shaped by insecurities. They were in a dialogue with other insecurities that demanded constant attention and anticipation. And those improvisations, in a way, themselves entailed new risks and new uncertainties. So I'm moving to the conclusion. So the West African outbreak of Ebola, like the COVID-19 pandemic that followed, was marked by uncertainty about zoonotic origins. Research on pathogens carried by bats has intensified, and this has given a special place to scientists and field workers in the regions where biodiversity hotspots overlap with disease hotspots. Across Africa, also Southeast Asia, researchers have been recruited in 2 surveillance projects aimed at identifying viruses circulating in wildlife before they emerge in humans. Now, in public representations of this work, which we saw earlier, representations of Maganga, for example, African virus hunters have become symbols of preparedness. Media accounts celebrate their courage, their ability to operate under difficult conditions. Global health institutions highlight the resourcefulness of those workers who sample thousands of animals in challenging environments. Such portrayals obscure the conditions that make improvisation necessary in the first place. This presentation has examined the labor that lies behind those representations. One Health surveillance seeks to balance human health and animal welfare. Research protocols attempt to reconcile biosecurity and bioethics by prescribing procedures that are safe and humane. Yet the work of implementing those principles falls to field workers operating in contexts marked by infrastructural fragility. For the samplers of PREDICT, sampling bats was never simply a matter of following protocols. They sought to protect themselves, care for bats, satisfy scientific requirements, also preserve their livelihoods and maintain dignity. And the result was not always a stable balance between competing values. These aims did not always align. Sometimes compromises were possible, sometimes they were not, and bats died along the way. or field workers were bitten. They had to anticipate equipment failures, animal injuries, transportation difficulties, shortages of fuel, misunderstandings with local residents, managerial scrutiny. They had to navigate expectations about professionalism, masculinity, and self-sacrifice. Fieldwork required constant assessment of uncertainty. Preparedness really emerged through ongoing adjustment among these multiple vulnerabilities. So, put very simply, the labor of virus hunting is not so much about controlling uncertainty than working with it. The real infrastructure of pandemic prediction is found in such practices of adaptation and anticipation. It is found in these people who continually sound out vulnerabilities, human, animal, technical, and institutional, in order to keep surveillance moving forward. And in a way, they do like bats who navigate obstacles through echolocation. So just before concluding, it may now have become clear why the book is titled Echoes of Ebola, because the book tries to rethink pandemic preparedness as a dynamic anticipation of uncertainties, including those generated by the work of epidemic preparedness. And to elaborate on this dialogical dimension, I use a model of the echo and draw inspiration from a mode of navigation that is not human, but the navigation of bats through echolocation. To the zoologist Donald Griffin, who coined the term echolocation, this skill is inherently different from other more passive senses, because bats do not merely extract signals from their surroundings, they actively emit signals which ripple back, are received and interpreted before the bat adjusts its course. To recognize an echo, one needs to hear the initial sound and the way it bounces obstacles. And like the bats even produces echoes, interpret the delay between sound and echo, and draw conclusion then about its movement vis-a-vis the source of the echo. By analogy with the echo, we can model anticipation as a dialogue of science that reflects and helps subjects, like bats do, grasp the coordinates of their own moving position. Epidemic preparedness is an endeavor where viruses that may arise are made present through the back and forth between institutions, lab, country authorities, and the laborers of global health. When scientists study the pathogens that may cross over from animals into humans, mostly they vow to reduce the uncertainties about the mechanisms of transmission and the potential of those diseases to trigger a sizable epidemic. This is Frédéric Keck, for example, who has talked at this seminar, has been investigating these this threshold of epidemic preparedness, separating humans and non-humans through looking at the sort of signals that bad viruses send us. But this book is looking at other thresholds of epidemic preparedness where other uncertainties are mitigated through other signals. Virological surveillance, indeed, takes place on the edge of social, ecological, and institutional worlds pervaded by moving and opaque unknowns that cannot be foreseen and need to be relentlessly sounded out. And I thank you very much for your attention. I hope I made it in time.
Thank you, Emma. Yes, you did, and thank you so much for this marvelous talk. I'm talking, but I see many people clapping. It was an amazing talk. Thank you so much for bringing us to this world that we often do not know of, animal sampling with its material and affective dimension that you've described so well. So many people might know this, but I know Emmanuelle's work very well, and so I will ask some questions if we have not eventually. But I also want to encourage colleagues, especially veterinary colleagues, to raise their hand and ask questions if they have. I think this is like a great opportunity that we have in the seminar to bring together different disciplines so we can have a conversation together. So if you prefer to have your question in writing, I'm also happy to read it. If you think your micro is not reliable and you prefer to have it writing, it's also fine. You can also use the raise your hand Zoom function, but I see that we already have one question in the chat from Yash. Thank you so much, Emmanuelle. I would love to know if workforce surveillance technologies have a presence in projects such as Predict. If given the choice, would supervisor wield such technologies to extract greater protocol compliance from sampler in the field, like limits the improvisation?
Yeah, thank you, Yash, for this great question. So if I understand correctly, you're wondering whether the supervisors or like the contractors, mostly based in the US, how they were trying to kind of to monitor and surveil that work. And you are absolutely right. So it's a work of epidemic surveillance, right? But the laborers themselves, that work themselves is very much in the limelight because it may cause so many risks. And so a lot of the technologies such as spreadsheets, for example, such as photographs, as we've seen, were also used to, it's interesting because they were used, I think, to indeed verify compliance with protocols. So they were observed, photographed, for example, those pictures, they were observed to see whether samplers were respecting biosafety measures. But I think the samplers themselves were anticipating. I mean, I'm not arguing that the US contractors were necessarily completely aware and using those technologies as surveillance. What's interesting is that the workers, those field workers, are very much used to accommodating expectations from distant funders and navigating them, and so they always feared being kind of taken caught red-handed, and so I'm not even sure whether all the photographs were carefully looked at, but the samplers in the field really feared the power that those photographs might have in the US, and if they were caught making mistakes, they could be used to then terminate their contracts. So it's you are absolutely right, I think, to assume that those technologies were used, and it's interesting because they were not only used, they're also expected to be used.
Thank you, Emmanuelle. Perhaps if you can have a quick look in the chat. Also, we have some questions for Matthias Bisong. I hope I'm pronouncing well. Perhaps so he's asking how was sample storage was handled and transportation. Perhaps that's a good thing if you explain the anthropological nature of your fieldwork and the ethnographic study you've conducted, and the fact that you're not a veterinarian, sorry, and you've not sampled bats, but you've studied this whole process. So I let you take these ones.
Yeah. Thank you, Matthias. Yes, just to be very clear, perhaps I could have been clear at the beginning. I was not paid by the Predict project. I do not represent the project, which has been terminated anyways. I was actually quite generously allowed to take part in missions, but I'm not a vet doctor myself. I'm an anthropologist, and I kind of navigating my position between mostly the samplers, the supervisors, the US scientists, mostly without trying to interfere too much with the work that was being done under very difficult circumstances. So I was also trying not to add to their burden already. So I'm not an expert. Sample storage, I mean, it really depends on what virus hunting project we are looking at. That project in particular had a very kind of elaborate protocol. I said they collected 9 samples per animal. At some moments of the project, they were using different collection mediums, some that kept the virus active and others inactivated. And then in theory, the samples that were inactivated would be transported to the US. They were being stored on in the field site in nitrogen tanks. When those were available, quite often fieldworkers also had to improvise around that. So they would use like plastic coolers that were could be quite broken. The cold chain wasn't always really easy to respect. And yes, they did do species identification. Again, because of the reliability of RNA identification in the lab, they did not bother much to identify the species in the field, which is also a particularity of this of these massive scale virus hunting project that I was able to witness, which is different from perhaps like smaller scale research. It was really like a 4-gist assembly chain, one bat after the next, and not a lot of expertise or time to identify sample bats. Knowing that you could use, or thinking that you could use RNA in the lab if you find a virus to then identify the species. So those are some details, but I don't necessarily not necessarily speaking on behalf of the project managers.
Thank you. We have a question from Soeyu. I hope I pronounced right.
Hello, am I audible? Yeah. Yeah, great. Thank you very much. I think it's a really eye-opening and great talk. I'm from Utrecht University at the veterinary medicine, and I think I want to hear a little bit more, particularly about the sharing of data. Because I think, as you are aware, now we are talking a lot about pathogen sharing and benefit. The whole pandemic negotiations are extended because the member state at the WHO cannot agree on that. So I think one treaty took place in Myanmar, where I'm originally from. And now I think 10 or 12 years later, a lot of things has changed as well. There's no funding anymore. There's no capacity to even do the real-time sequencing in the field. So in that sense, how are you reflecting yourself as a anthropologist on these things that happen? I mean, Congo, like now we can share the sequence very quickly, but in terms of preparedness perspective and power dynamic when it comes to pathogen genome data, what are your thoughts on that, and how can you predict for future as well? Because now nobody really has an answer on that. Thank you.
Thank you, Soye. That's an amazing question. I actually have a entire chapter dealing with this issue of data sharing. So obviously, one of the assumptions of these projects is that data is shared quickly and transparently and uploaded to databases. And actually, the West African Ebola outbreak introduced some changes for real-time sharing of pathogen sequences, so not, at the time, human sequences. It became kind of super common, I think, around that time to also upload preprints to repositories. So in order to inform the intervention in real time. But what I realized is that PREDICT was performed by USAID, right? So it was also development work. It was also high level scientific diplomacy, and they were also quite attuned to the necessity to manage some space for state sovereignty. So I'm I have a chapter that's dealing with the way in which Guinea was trying to assert its viral sovereignty, because obviously those pathogens can be used to inform interventions, but they can also generate vaccines and treatments. And there's this the Nagoya Protocol I think it's like 2011, if I'm not mistaken, on data sharing and access to benefits that kind of regulates these. I said regulating that sharing to ensure that like nation states that provide the material also benefit from it. Just to just sum up, what I could observe in this particular case is that the states, in the case of Guinea, with an interesting post-colonial history, was not so much interested in asserting sovereignty over the material samples, over the blood itself, if you want, because of the fear that if people knew in country that there were blood of Ebola patients or potentially infectious bat samples stored in the fridges of the National Hemorrhagic Fever Lab, that it would create, incomprehension, anger, trigger rumors that the president, which had been very much criticized at the time, that he was collaborating with foreign countries to trigger another Ebola outbreak. So the country was kind of like happy to get rid of the samples, but what they really wanted to have control over is the information, like when is the information released in country that new viruses were being discovered, such as in West Africa they discovered a new species of Ebola virus, the Bombali virus, and yeah, so this is to just like to make a very complex story very short, but it's a story where, as you may know, the CDC was involved. The Defense Threat Reduction Agency also was involved in controlling national labs, and it's really kind of as far as I could ascertain, it's a story which is really high politics, and embassies and presidents kind of actually can weigh in on the issue of whether those samples and information are being circulated or whether the information is blocked, and a lot of information is just also not released.
I will look for the book, I think. Read in your chapter. Thank you for sharing. Yeah.
And thank you for the question, Zoe. And when you first talk about data sharing, I thought the question was going in a different direction, and you were going to ask about the accessibility of Emmanuelle's work, and she's not going to tell you herself because she's too humble, but dissertation is open access. So if you want to, all of participants, if you want to have a look or perhaps go more in depth on a matter she only briefly touched on in this 40 minutes, I have sent the link in the chat. You can also obviously have a look at a Google profile and the other publication, but and obviously buy the book when it's out in a few. But so Maddie, you had raised your hand up.
Can you hear me?
Yeah.
Yes. Oh, so thank you very much for the presentation. It's really nice. I look forward for the book as well. But my question is related to the tension between biosafety and conservation of bats, because at least here in Europe we have this kind of regulations that prohibit to kill bats because it's also an animal that prevents control mosquitoes. And then how in Africa and global health, and then the epidemic of Ebola, how this biosecurity narrative came through, conservation narrative came through the tension between biosecurity and conservation.
Sorry, I did not understand the question.
If there is some other narrative than biosecurity that mostly came from after to capture the bats and like test them for checking if they are free. If there is also how the narrative behind also biosecurity and conservation, because bats are also useful in the ecosystem. So how they bring this narrative to this frame.
Yeah, great. I think I understand now. Thank you very much. So yeah, to be very clear, I mean, so Predict was a One Health project and was very much also committed to not killing animals. I mean, they sampled, I can't remember, I think tens of thousands of bats around the world in total. So it would have been unimaginable to kill every single one of them, which is, with smaller projects may sacrifice animals, because there's another balance that I didn't touch upon. But when you are sampling bats' saliva or urine, you may see certain pathogens. But if you can have access to the animal's liver or the spleen or the lungs, you may see you may just have access to other pathogens or to viral RNA, which is not being shed in urine. So there's a balance to be found as well between the goal of biosecurity, of discovering of new pathogens, of ensuring epidemic surveillance, and bat welfare. And I've definitely observed other projects that do not shy away from sacrificing every bat that they capture. But yes, you're absolutely right. Predict was very much committed to this idea of not wanting to vilify bats, and of also acknowledging that they contribute to the environment through dispersing seeds, through pollinating, through controlling mosquitoes. And so they also there's been a lot of One Health communication tools that were developed also in order to raise awareness in the residents of these places about the benefits of bats, the benefits of bat population, and the risks that these populations can also have. And so they were always highlighting the distance that should be kept with regards to bat populations. Distance which is not, in many cases, not really easy to maintain in these places. But there was another narrative, but projects such as those have been criticized a lot by conservation organizations for overly focusing on the dangers of bats. Also the imagery, some of the pictures that I showed you, they are, the bats appear very threatening, very dangerous, they pullulate. And that's, I've worked a little bit on the sort of imagery of bats that is circulated by conservation organizations, which is very different sorts of images of bats and bats workers. Also, if you're a conservation scientist, you're likely to wear a lot less protection than if you're a biosecurity scientist. So this is thanks for bringing that up.
Thank you.
And okay, so we have multiple questions in the chat. Emmanuelle, back to Louise Renaud's question. You document the insecurities that permeate the labor of virus hunters, avoided sites, strict PPE, hidden activities. But how do these insecurities systematically bias the data itself? And if so, what does that mean for the models and policies built on top of it? And what would it take to make those bias visible and correctable?
Excellent question. These insecurities absolutely become integrated in the data and bias the data, if I may say. I can give you a few examples, because samplers are afraid of sampling bats in the place where they were collected, especially if this place is a village and you do not want to be found sampling bats in the middle of a village at night because you are wearing protective equipment that looks very threatening, and so you don't want to be exposed to people's anger and attacks. They would be carrying the bats away, so leaving them in the bags, taking them to a distance, sometimes putting the bats in the vehicle itself. So in that process, the bats could contaminate each other. So you could have some dangers of cross-contamination. Potentially, you would also increase the risk that the bats contaminate the vehicle itself. So that's one way in which, for example, the data was affected. There would have been, or for example, the spreadsheet, the way the spreadsheet were being filled, the samplers felt that they had to like bring bats from villages, from the forest, from the fields as well, but they would sometimes just like fill the sheets just in the way that was most convenient, just to make sure that they were matching the expectations. What does that mean for the models and policies built on top of it? To be honest, I think those massive amounts of data, there's not so much policies and models that were built on top of it, because the data wasn't in a way, so it wasn't very much tightly integrated with fine-scaled ecological work on the practices that may expose people, or on the cycles of bats immunity that, you know, and the different seasons, and how viruses virus load kind of may evolve in a bat population from one season to the next. The prescriptions were very gross, like very general, you know. They would still kind of stay away from bats. So in a way, you did not even need like biased or flawed data. The data was just, in a way, a performance in order to like to run these huge communication campaigns that didn't need really the scientific work in the first place. So that would be kind of my response to your question. And what would it take to make those biases visible and correctable? I think, and that's kind of what I conclude the book on. I mean, a lot of that virus hunting work has been banned or halted actually with the COVID pandemic, and suspicion that it may have created more outbreaks, or may have led to a lab leak and to the COVID pandemic. But I think that the idea that you can just reform that sector with more surveillance, or sending better experts, or even increasing wages, is a bit of a fantasy, because this work is so tightly connected to the insecurities that permeate these places and make outbreaks possible in the first place, that it's just impossible.
Thank you, Emmanuelle. So we'll finish on time, because Emmanuelle has a train to catch. So at 13:55, 55, yes, we'll finish. So we'll take the last two questions together. So there was the first one by Professor Moabit. I would like to ask whether there are significant differences between the 2014 outbreak and the current outbreak in terms of epidemiology, transmission dynamics, and disease burden. Furthermore, how has the One Health approach contributed to the control of the outbreak and the improvement of the overall response? Before you answer that, Emma, I just want to remember everyone that Emmanuelle has conducted most of her fieldwork for a PhD thesis that she presented after the epidemic outbreak and out of epidemic response time. And so there was another question from Yash. Did viruses exist as distinct entities from other microbes in the public imaginary in Guinea pre-Ebola, and following the outbreaks, are they now considered distinct entities? So, Emma.
Okay, I think my answers will have to be brief. Thank you, Professor Mourabit, for your question. I'm afraid I can't really comment on it. Julien is a much better position to do so. I have not been working so much about the current outbreak. Obviously, we're talking about outbreaks. It's over 10 years since the beginning of that first outbreak. There's been a lot of I mean, there was no vaccine back in 2014. It's a very different region in terms of civil conflicts and transportation. The West African region is a different environment and is very well connected. Another key difference is that since 2014, the immunology and symptomatology of Ebola have been very much clarified, and I think communication has very much changed as well. So back in 2014, a lot of the advice was focusing on bushmeat and funerals. I mean, Jules, you correct me if I'm wrong. I mean, we still see those, but we're also a lot more aware of the key role that survivors play in resurgences of the disease. And so it doesn't look like the current outbreak started with a survivor or someone that's been infected by Ebola, Bundibugyo earlier, but it's there's a lot of differences as well in the way responses are being carried out, and in also the big communication campaigns around them. So I couldn't comment more on the One Health approach and to this current outbreak. And just, I mean, Jules, you may want to say something.
No, you're fine. You're fine. We'll have a session dedicated to the current Ebola outbreak in DRC at some point. So it's better to. This is why I started by saying, okay, you conducted your ethnography out of outbreak response time, and so we really want to hear from you and on this situation. But if there was.
Yeah.
Well, we've discussed that together already. Also, there are comparisons to make also, but we really want to focus on your work in this session, because, as I said, we'll have others dedicated specifically to this one. Emma, you freezed? Or is it me, Paola?
You're right. She's frozen.
Okay. Sure.
Yeah.
Okay. Well, we can leave her some minutes, but yes, thank you, Clément. I'll just check with her on WhatsApp, and otherwise we'll call it a day. But let's see if she's coming back. Anyway, that was unexpected, but she'll be back. She'll be back in no time. Well, so we have that's also the opportunity while waiting for Emmanuelle to come back to announce a season 2 of the seminar in the next academic year. So keep positive and have a look at. There is a mailing list. There are plenty of different. So also feel free to email me if you've not heard anything by early October. Just be in touch, and I'll send you all of the material. But it's a good thing it's happened at the end of the seminar.
Yes.
We've had
but it was great. It was great. Very sharp, very dynamic. You see a lot of interactions with the audience.
Yes, something else I wanted to say is that when we designed the program, that was really also a session because there are many different partners as part of the graduate school, and there is a veterinary school at Maison Alfort in France, and this whole idea of having a One Health initiative that was a little bit, let's say, the session more dedicated to aiming specifically at veterinarians and examining really the animal sampling, but as an entry to more broadly interrogate animal health questions. So I was really happy to see a lot of veterinarians in the call.
I don't know if Emmanuelle is coming back, but I would like to thank you, Jules, because it was a really good webinar, and it's helping us to understand how society produces knowledge about infectious disease risk, how preparedness is organized, how scientific practices are shaped by social, political, economical, and material conditions, and thanks to this presentation made by Emmanuelle, also by ontological conditions. So thank you very much for helping us connect with this amazing group of researchers, for bringing together also the students, the researchers. It was a really nice series. And I see you are discussing me with her.
Pardon, sorry. Yes, she was just on the phone with me. So, well, I was hearing two conversations, but well, thank you very much for what you said, Paola. I caught a few things. So yeah, Emmanuelle called me. So Wi-Fi issue, classic. But so by the time, well, she couldn't reconnect, and by the time she could reconnect, she needs to leave and take a train anyway. So we'll call it a day. She's very sorry. She was thanking all of you for your questions and this very dynamic webinar. It was really a pleasure for her and for me. And yes, we'll see us for the next season in the next academic year. Bonnes vacances tout le monde, and goodbye. Paola, you wanna add something or?
Well, I would just say thank you again. Thank you to all the participants for your commitment and enthusiasm. It was really nice to have you every month sharing with us your questions, your comments, saying hi from different places around the world. We hope to see you next next academic year. And for those who are still interested, we have on the One Health Research in French Amazonia next session on June the 24th. Thank you very much. And thank you again, Jules.
Goodbye. Thank you.